Oh, you hear me, guys?
Talk. We hear you.
Okay, perfect. So I think I'm not allowed to turn on video right now, but that's okay. That's okay. Um, okay, if everyone is in, then we start. Um, I welcome everyone. Sorry? Is Further actually here? Because I don't see him in the um, I'm on the online participants right now.
How old is Sissy?
Stacy is a baby, and he's great.
Wow.
So if she gets married to your brother, they're good.
Wow.
Money, right? Money, right?
Money won't be a problem.
Because Sissy can't suffer.
Money won't be a problem.
She needs to enjoy her life.
Please mute yourself when you join. Please mute yourself if you join. Check if you're muted, please. Yeah. Okay, I hope we will be able to start in a couple of minutes. I'm sorry for confusion and delay. I just was able to join this video and then it said the host has stopped your video. So I don't know. I could be that the reason, but I mean it's okay for me. I'm just gonna chair, but for speakers maybe it would be good if they can have video on. Yeah, so we have a delay. We need to wait for speaker to connect.
Hi, everybody. Sorry I'm late. I was convinced my talk is in 2 hours.
Really, so for the confusion, will you be able to give a talk now?
Sure.
Yeah, okay. Thank you.
No, sorry about this. It's time is hard. Let me just find the file.
Yeah, and while you're looking, I will introduce briefly and then we can go. Thank you.
Thank you so much.
Yeah, all right. Welcome everyone to day 2 of Ukrainian School in Evolutionary Biology. So today's first speaker is Fyodor Kondrashov from Okinawa Institute of Science and Technology in Japan. He studied in Eugene Koonin's lab, then graduated with PhD from University of California, San Diego. He's receiver of prestigious Theodosius Dobzhansky Prize from Society of Evolutionary Biology, and also M. Boyan Investigators Award. So please welcome Fyodor, and the floor is yours.
Yeah, thank you so much, and sorry again for me being late. I want to give you a little bit of an introduction to very important problem that we study in a lab, and I want to do it in the following way. I want to structure my talk more about conceptual side of things, and a little bit less about actually what we do and what we care about. And because I want to sort of do this more, I guess, more conceptually, I will try to, again, sort of focus on this big blah, 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 blah. But please interrupt me if there's anything that is unclear. So let me just start this presentation. 1 second. Here we go. It will, so especially in what I want to talk about in the beginning, I think is actually quite important, because it's sort of it's the main thing that in our field people care about, and it's also the main thing that I think is conceptually important in the field. So hold on 1 second. Let me start the presentation here, and let me see how this goes. I need to share screen. Here we go. Yeah, you guys see this now? Okay.
Yes, we can see online too.
Thank you. So this is the main question. I hope I don't need to introduce what is a genotype or what is a phenotype. Genotype is just, sequence of DNA. Phenotype is whatever you actually see within the organs. So phenotype is actually a funny thing, and this is from an evolutionary perspective. Of course, what we care about as a phenotype is fitness. Fitness has many definitions, etc. But one thing I do want to impress on you is that fitness, because you can measure it by definition, it's also a phenotype. So whenever you hear a talk about similar things, when people go from genotype to protein function or something else, they're really talking about very, very similar things. Now, here is, with this kind of connection between genotype and phenotype, this is a much kind of a complex thing. What do I mean by this error? How do we go from genotype to phenotype? It's very difficult to become quantitative, or let's say theoretical, about just this description. And of course, this description is just used to depict what we care about. But we can put this into a more molecular basis. For example, we can start asking the question, does the sequence do anything? So any guesses that do these sequence do anything? Can you tell by looking at the sequence? People in the audience or people online?
One on the spot, so if anyone has idea or online, please raise your hand. I will try to.
No worry. Alexander, thank you. But the question, the reason why I'm asking the question is actually really hard to tell what this sequence does, and that is exactly the point, right? It is really a difficult question to answer, and and I can give you the answer. The top is just a random sequence I invented, and the bottom is a green fluorescent protein from Macrorhiza victoria. And you can see that the very little differences between them. You won't necessarily know which one is which just by looking at this. And actually, the point I want to impress on you, it's so difficult to tell them apart that I actually just lied to you. really, it's the top one that is actually the GFP sequence, and the bottom one is random. And again, the point is that you really would not be able to tell the difference. And of course, I just lied to you again. Actually, the top really is the random sequence, and the bottom is the GFP. So how would you really know? Like, am I lying this time, or did I lie to you before? And this is a very, very important part about our field. What we would know what a sequence does is not really by looking at the sequence, in this case, the genotype itself. The way we usually work in the field of, this kind of a, let's say, evolutionary or molecular evolution, we would usually use information that we already know about the sequences to make sense of it. You would not necessarily just look at the random sequence. And of course, the way you would do this in this case is you would compare this sequence to other existing sequences. Right? In this case, this is a tool called BLAST. Also, I see you guys in the audience. Please raise your hand if you heard of BLAST. Yes, very good. Some people are raising your hand. Thank you. So BLAST, right, is just a tool that allows you to compare sequences. And this is, of course, would be one way for you to tell whether or not the sequence does something. So these two sequences I just showed you before, you can take both of them, blast it, meaning comparing it to every other protein sequence that have ever been discovered, in mankind, in GenBank. And for one of them, this is the GFP, you will get a really good, what's called hit, right? A really good comparison. There's a lot of amino acids that are similar between what I've given to as a query and the result that I got. And of course, this turns out to be a GFP. And if you were to take a random sequence and do a blast, you would get nothing. You would get no significant similarities. So the point being is that actually pre-existing knowledge is so far our best and more or less only tool to be able to tell that a sequence actually does something. And that is actually kind of depressing, right? So we can start exploring this genotype space a little bit. For example, let's say we introduce a mutation into GFP, right? We already know that this is GFP, either because biochemist has characterized it and, they extracted this protein and they can show that it fluoresces, or that you can do this by...
моей дочке.
Sorry, you can do this by comparison. But the question is, okay, so what if you introduce a single mutation? Does the sequence still do anything? It's a good question. We are okay in solving this problem. Not necessarily for GFP, but for many other proteins, especially human proteins, especially if we care about not fitness, if we care about disease state. But we have become good enough to be able to tell whether or not one mutation still keeps the protein functional or this mutation will break the protein. We are really bad in solving the problem whether or not two mutations break down the protein. We are, I would say, impossible in answering the question whether or not 10 mutations or 15 mutations keep the protein functional or not. And again, the reason why I'm showing you all this is to actually drive home 2 points. Point number one is that when we ask more or less any question that you can think of in biology, for example, does this protein function? What is the effect of an enhancer sequence? What causes a disease? In essence, this is a question of how do you go from genotype to phenotype? And from that perspective, it is the most fundamental question you can ask probably in biology and for sure in evolutionary biology. This is kind of a general thing. And two, the point I want to drive here is that this question, even though perhaps it's the most fundamental question in at least in evolutionary theory or in evolutionary biology, we're still not even close to actually answering this question. Now, there is a way to start thinking about this problem in a way that people have thought for a very long time, and I will get to this. The way we start thinking about this is trying to imagine not just a particular sequence, right? Essentially, when I show you a sequence of GFP, this is just one sequence, right? But you can start taking a look at diversity of different sequences. And when we start assembling diversity of sequences, we can call this the sequence space, or we can call it the genotype space. there's a subset of this which is called the fitless landscape, and I will get to this. So a sequence space or genotype space is essentially a collection of many different sequences together. Usually, if you're talking about the entirety of this of the genotype space, is all of the possible genotypes of a certain length. So this is here a representation of a genotype space of the length of two nucleotides, right? So there are two nucleotides, so there's just a 4 to the power of 2 different possible genotypes. Now, I'm turning this into a fitness landscape. And what I mean by this is that when for a particular genotype, I actually know what is its phenotype of interest. In this case, again, fitness. So here there are genotypes that have very low fitness. These are genotypes outlined in black circle. There are genotypes that have high fitness. Those are genotypes that are outlined in the green circle. And here I have some set of rules that allow me to determine which genotype has high fitness or functional, let's say, and which genotype is non-functional. If a sequence has a G or an A in the second position, it means that that sequence has low fitness, and everybody else is happy. So essentially what I'm introducing deleterious mutations, or let's say disease-causing mutations that break the function of the sequence, and then everybody is fine. So there are very, very simple rules by which I determine what works, what doesn't. You can imagine that those rules are very complex. So here's another fitness landscape where I have the same fraction of genotypes that are fit or fraction of genotypes that are not fit. Half of them are fit, half of them are not fit. But the rules by which I created this landscape is very different. So for example, here you have a genotype. I hope you see my mouse. You have the genotype of AG, which is not fit. And in the previous landscape, AG would have low fitness because G is this deleterious state. But here you have another genotype TG or another genotype CG, which has a G at the second position, and this is perfectly fine. So therefore, this genotype of AG is bad or has low fitness, not because it is itself bad, but because the interaction between A and G somehow created bad. So this interaction between two sites is called epistasis. And this is essentially what makes life interesting. Because let's say if you're starting in this original non-epistatic landscape, you're starting to see an evolutionary trajectory from AT into GC, you can do it with just 2 steps, right? From AT, you can become AC. From AC, you can become GC. Very easy. Or you can go through the GT intermediate. In the second epistatic landscape, you can still do it. You can still go from AT to GC. It's just now the shortest path that is available to you is not 2 steps, it's 4 steps. So, and that I think demonstrates quite vividly, I hope to you, that whether or not interactions between sites are important in determining how you go from genotype to phenotype are important, this really affects the evolutionary process, right? In this case, the evolutionary pathways between those 2 genotypes will become twice as long. So this is kind of just to give you a taste. Now, the first person that has described fitness landscapes or thought deeply about how genotypes go into phenotypes was a guy called Sewell Wright, who in 1932 wrote kind of a review of the models that he was trying to do, which is now the paper that everybody cites as having kind of fathered this idea of the fitness landscape. I will spend a couple of minutes describing this slide. And I kind of, every time I talk about this, I'm kind of in awe of the brilliance of this guy, because, he was working in the late 1920s, early 1930s. Nobody knew anything about sequences. People did not know that genetic material is the DNA. Nobody had any clue what proteins are. They knew that proteins existed, but, very few, or if any, protein characteristics have been described. Because he was a geneticist, right? I mean, they didn't really talk about sequences and sites and amino acids and nucleotides. They talked about loci and they talked about alleles. But from this perspective, you can think of loci as sites or loci as genes, and of course, alleles as variants of those genes. So you can think of alleles as different amino acid states in those sequences. So in this case, Sewell Wright, again, nobody knew nothing about genomes. And he said, well, okay, there is probably in a big genome, like in a eukaryotic, in a complex genome, there's probably something on the order of 100,000 loci, genes or sites, that have different alleles. So he said, okay, well, if you have only 2 loci in your genotype, right, and you have, here's your wild type. This is what you're starting with. This is the GFP that I showed you, the original wild type sequence. If there are 2 loci, you can have relative to the wild type either a substitution or an allele change in one locus, an allele change in another locus, or both of the loci can be different from the wild type. Again, because he did not know about nucleotides, he assumed that there were just 2 different alleles per loci. Now we know per site there's at least a nucleotide size, there are 4 different nucleotides, right? So he said, okay, well, this is a 2-dimensional space. If there are 3 loci, meaning 3 interacting genes or 3 sites, well, all of a sudden this is a 3-dimensional space because you can have combinations of different alleles in 3 different loci. If this is if there are 4 loci or 4 sites in your genotype, this is a 4-dimensional space. If there are 5, there's a 5-dimensional space. If there's 100,000 genes in your genome, that means you're dealing with 100,000 dimensional space. Now, Sewell Wright was very smart, but he recognized that it's actually very difficult to think in 100,000 dimensional space. I have difficulty finding my way to work in a 2-dimensional space of roads in Okinawa. No? So, 100,000 dimensions is even harder. And a lot of intuition that we have for thinking how do we actually traverse spaces in 2 or 3 dimensions may not necessarily work in 100,000 dimensional space. Now, Sewell Wright has recognized this, and he said, well, okay, let's think about fitness landscapes, or let's think about, let's say, genotype spaces first, in a different way than just starting to think in this very highly multidimensional space. Perhaps we can somehow condense the complexity of 100,000 dimensions into 2 dimensions. So what this diagram is, and this is figure 2 from his paper in 1932, is a representation of this very, very highly dimensional and complex space in 100,000 dimensions. But all of those genetic dimensions are represented just by 2 dimensions, the x and the y axis here. And the height here, the pluses and the minuses, this will be like a topographical map of, let's say, the Carpathian Mountains, right? So you have mountains that are pluses and you have valleys that are minuses. And the height here represents fitness, which genotypes are fit or not fit. So his point of that paper was that if somehow it is feasible to reduce the complexity of all of the genotypic interactions of 100,000 dimensions of your loci into just 2 dimensions, it becomes very easy to have an intuitive understanding of the evolutionary process. What that process would be somehow wandering through the genotype space in those 2 dimensions, maintaining high fitness and avoiding areas of low fitness. And we know that this to be the case because, by just my classical population genetics, we know what is the probability of the fixation of deleterious alleles. It's actually very, very small. So we know that an evolving population actually maintains high fitness of individuals in that population. So, and then, essentially, so this is kind of, I want to stop here a little bit to see if you guys have any questions, but to recap a little bit, Sewell Wright created this abstract mental concept of the fitness landscape, which is akin or very similar to a topographical map, where the rules of the game is that a population, when it changes its genotype, meaning that when it evolves, what it does is it keeps high fitness and it evolves in the genotype space, only exploring those areas of genotype space which have high fitness, avoiding those areas of the genotypes which have low fitness. And again, this is a toy model that it's, in 3 dimensions, it's easy to think about this. In 100,000 dimensions, the rules will be exactly the same, but it's just very difficult to think about in 100,000 dimensions. So when people usually draw these fitness landscape, they tend to stick to 2 dimensions. Any questions on this part yet? Good. If you guys do, please interrupt. Now, here's a problem. There are several problems that we have, I think, conceptually in studying fitness landscapes. Now, I told you that all of the problems in evolutionary biology can, to some degree, be reduced to the questions about fitness landscapes. We still, to this day, know really nothing about them. For example, one of the questions that, I think Sewell Wright made a mistake actually in drawing this picture, because he drew this picture by having isolated fitness peaks. Right, you can see that here's a mountain, here's a mountain, here's a mountain. But because we know that evolution really is possible, we know that real populations actually really do change genotypes. These three mountains should actually be connected to a single ridge, whereby you can go from one to the other. And because I think he made this mistake in drawing this picture, a lot of the field for a while has been, asking the wrong question, which is, how do you jump from mountain to mountain, which is actually not really the case, which is, you go from mountain to mountain across a ridge. Another question, so here you have this one big area of low fitness here, right? So there's minuses and minuses here. Well, maybe there's small little mountains that you just don't see here. So there are maybe some peaks there that haven't been identified. A more conceptual question that, he drew this picture, assuming that it is possible to reduce the complexity of interactions from 100,000 dimensions into 2 or 3 dimensions. Maybe this is not the case. The point being is that empirically, we know practically nothing about the general rules of how you go from genotype to phenotype, which is exactly to say the same problem that I showed you before. Given a sequence, we really don't know anything about what it does or not. And this is where I want to talk a little bit about what we have done. So we've been working with GFP. So GFP did not come randomly in my example in the beginning. And we decided to actually start exploring the fitness landscape of the GFP. Now, in this case, I'm calling it the fitness landscape, even though what we are actually looking at is whether or not the GFP fluoresces. So it's a function. But this is, as I told you, because fitness is also a phenotype, essentially, we're just looking at the genotype to phenotype connection. And the way we're doing these experiments is the following. We have the wild type. This is the plus in Sewell Wright's representation, right? It's a GFP that fluoresces. And by introducing experimentally just random mutations into it, we can start looking at phenotypes or fitnesses of single mutants, double mutants, triple mutants, and essentially it's a matter of what is the function of the random introduction of mutation into the wild type, of how far away from the wild type we go. So it's a characterization of what we call the local fitness landscape, which is essentially just a fitness peak, right? It's like we're looking at a mountain, we're sitting on a high mountain, and we're looking several steps away from that point in space. And we can do this experiment relatively easy, right? Because GFP has a very nice phenotype. You can express a GFP in a bacteria. You can sort this bacteria in a cell sorter, and you can determine the level of fluorescence of that GFP just by sorting a bunch of mutants. And here's one of the representations of what this fitness landscape looks like. Now, because we go up to about 10 mutations, what I'm actually showing you is 10-dimensional space. Now, as Sewell Wright has recognized, it is very difficult to draw 10 dimensions in a 2-dimensional space. So we are stuck with various ways to start to try to represent this complexity in a 2-dimensional setting. One way to do this is to look at the distribution of functionality as a function of the number of mutations in your genotype. So here in the beginning, right, it's just the distribution of the wild type. So essentially what this is our error rate of how accurately we can measure fluorescence of the genotype. Now here is the distribution of the level of fluorescences with a single mutants. So you can see that a bunch of single mutants just kill the fluorescence entirely, right? So these guys are completely lethal. But you can see that this distribution of fluorescences on top here is actually wider than our distribution of wild types. So therefore, we capture some fraction of alleles or mutants that actually decrease the fitness by a little bit or decrease the fluorescence by a little bit. And here, here's 2 mutants, 3 mutants, etc. And by the time you have 5 mutations, more than half of your genotypes are lethal. More than half of your genotypes no longer function in any way. So this is another attempt to represent this fitness peak. This is a drawing of the protein with the head of the protein connected to the tail in this kind of in a circle. And then again, the width of the circle represents the number of mutations that this genotype has. And the surface here is the median level of fluorescence of all genotypes that have a mutation in that particular site, right? So it's trivial when you're talking about a circle that is just with one mutation. There's only one mutation in that protein, right? So that will be the surface will be, right, the protein that has a mutation in that site. Well, if you're talking about two mutations, right, you're talking about this genotype here, one of the mutations will be in that particular site, and the other mutation will be anywhere else. So this is also really even though this is a three-dimensional representation of the fitness peak, this is exactly the classical Sewin-Wrightian fitness landscape, because I'm essentially making an attempt to compress 10 dimensions into 3 dimensions by averaging out the impact of interactions of mutations between different sites. So again, this is a representation, and again, perhaps not very accurate, and I will get into this later. So you have here the wild type in the center, and you can see that you're decreasing fitness a little bit on average, and then by the time you have 5 mutations, you're just dead, right? So it's a very sharp fitness peak. Now, I don't want to dwell on this so much anymore, because when we did this back about 10 years ago, this was revolutionary. This was the first fitness peak that was ever assayed experimentally on a very, very big scale. So we are dealing with about 60,000 different mutations that we have looked at, right? This was an order of magnitude more than anybody has done before us. This is not really that revolutionary anymore. People do fitness peaks. They're called deep mutational scans, or sometimes they're called, massively some, blah, blah, blah. They have another idiotic name for it. But so this is no longer really that interesting. And what we found out is that it's very easy to break a protein, and those fitness peaks look relatively sharp, right? As you start accumulating mutations, random mutations, you tend to fall off this peak quite quickly. Now, what I think is actually more interesting is starting to ask questions: do different peaks in different areas of the genotype space look different from each other? So essentially what we have done to reveal this is look not just at a single wild type GFP, but because GFP is found in many different species, not just actually in invertebrates. Some vertebrates, or at least tunicates, have them, and they're found not only in jellyfish, they're found in crustaceans, etc. They found really kind of across in many different aquatic sea species. We selected 3 additional GFPs to compare, right? And we thought we were very, very clever in selecting the GFPs that we were looking at. These are the GFPs. This is the distance between the protein sequences that we thought, because one of the questions that we were after, we wanted to ask the question: can we predict whether or not a sequence is a GFP or not? Any arbitrary sequence. So what we wanted to really do was to describe the entirety of the genotype space in a way that would tell us a universal story about how this genotype to phenotype connection looks like in the GFP. So we thought, okay, great. In order to describe the entirety of this space, we need some very, very close sequences. So we took this Ecrioria victoria. This is Ecrioria macrodactyla. There's only 18% divergence, so 80% identical. We took another jellyfish, which were 40% identity, so kind of a median distance. And we took a very, very distant GFP from a crustacean. So we thought, oh, okay, by understanding how GFPs differ from each other across various distances and genotype space, we will understand this entire genotype space. I will skip the experimental part, except for one part. So we have a way. So the way we do this is that we just very quickly we take the wild type GFP, introduce mutations into it, and we introduce a barcode, which is just a random piece of DNA together with this GFP. We sequence it all together, and therefore we have a one-to-one connection between the genotype of the GFP and the identifier, right, the barcode. After which, this library that we have now sequenced, we stick this into a vector, where the GFP is merged into a single protein with a RFP, so it's a red fluorescent protein. And the reason why we do this is because when we are looking at a cell and the cell either fluoresces or does not fluoresce, we want to understand that this is the function of the protein and not the function of the expression of that protein. So what we do is that when we sort those cells, we first sort them by their red fluorescence, controlling for the rate of the expression of this construct. And then we only take a very relatively narrow band. This is the 8A picture here. We take a narrow band of the red fluorescences of cells that express, GFP expressed at a certain rate, and resort only those cells, and then look at the level of green fluorescence of those cells. So that is a way for us to cleverly control the rate of expression, being able to target whether or not the genes are actually, the protein themselves are actually functional or not functional. And after this entire sorting of the green fluorescence, we don't need to sequence the entire gene. All we need to do is to sequence the barcode because we have this one-to-one association of the genotypes to the proteins. So anyway, this is what it looks like. This is the data. This is also a representation of all of the genotypes that we have done for all of the 4 different proteins. The yellow one, this is the one that we've done in the original paper. And what I want to show you here are 2 things. First of all, that this data is very, very accurate. So the way we know this is that we can look at the width of the distribution of these white guys here, and this is the width of the distributions of wild type or genotypes that only have synonymous mutations. Synonymous mutations, we do a separate test, they don't affect anything. But anyway, you can see that this distribution of wild types is actually relatively narrow, so we can measure them correctly. 2, we can look at mutations that affect the chromophore of the GFP that we know a priori will kill the function of this protein. So, and we see that all of these guys that actually kill the function of the protein are on this left distribution, so they're not fluorescent, they're dark. So we know that our error rates are very, very small. And again, these are sort of the distributions. And yeah, I want to skip this. But so this is exactly the same violin plots that I've showed you before, where you have, the wild type distribution, the distribution of single mutants and so on. And you can see, and this black line here in those 4 different distributions is again the average level of fluorescence of genotypes with a certain number of random mutations. And this is where it becomes interesting. It's interesting because all of these different 4 proteins have different tolerances for different mutations. So again, the yellow one is our original one. You can tell, right? So between 4 mutations and 5 mutations, on average, you go from being light to being dark. In Clitia gregaria, this is the GFP that we took from the crustacean. It's even more dramatic. You go from being mostly light to being mostly dark, somewhere between 2 and 4. So you're actually about, twice as fragile. But when you look at Aquarium macrodactyla, this is one of the GFPs from this is actually the closest one to this guy. You can tell that it's actually pretty robust, right? That it goes from being mostly fluorescent to mostly non-fluorescent, somewhere between 5 and 7 mutations. So it takes on average twice as many random mutations to kill this purple protein compared to this blue protein here. So we already start to see some fundamental differences between the GFPs. But I mentioned to you that what we care about is the fitness landscape. And the way to understand the fitness landscape is what I told you in the beginning. How much interactions are there between different sites of the protein? Again, if there are no interactions, the landscape is very simple. This was the landscape I showed you in the very beginning, where the rules by which you break a protein are, you have a universally bad mutation, and this protein is no longer functional. So, interactions between those mutations is what's called epistasis, and there's a way to quantify it. So essentially, you can determine the effect of a mutation. The effect of a mutation is the difference between the function of your wild type protein and this protein that has a mutation. Let's call this mutation A. And how much epistasis there is, how much interaction there's between two mutations, is essentially the difference between the mutant effect of two mutations occurring in the genotype at the same time versus the sum of the two mutations independently occurring in two genotypes. So if those two mutations are affecting your phenotype completely independently, then their sum of their individual effect should equal their joint effect together, and therefore this difference of this two mutations minus the individual mutation should be zero. So you would have no epistasis. If, however, the effect of two mutations is stronger than the effect of single mutations, you would have what's called negative epistasis, right? So this E would be either positive or negative. So as you start to accumulate mutations, your fluorescence or your phenotype will start to decline faster than you would expect just by summing up the effect of individual mutants, right? So again, this blue line here, right, what would happen if just everything was additive, and this negative epistasis, this is what would happen if the joint impact of individual mutations is bigger than their sum of individual effects. And this is what we see in our actual data. So it's a slightly difficult graph to understand, but on the y-axis is essentially the fraction of genotypes that can be explained without any epistasis, right? So this is the on the x-axis, right? Is the genotypes as the number of mutations away from the wild type, and the line here, right, is what fraction of them can be explained without any epistasis. Now, of course, if you have just a single mutations, by definition, there's no epistasis, right? There's no interaction between anything, so everybody starts from one. Now, this guy, for example, this yellow guy, Clitia gregaria, by the time you get to 5 mutations, you need to have epistasis to be able to explain half of all of the genotypes that have 5 mutations, right? So this is a very epistatic landscape, meaning that there's a lot of interactions between mutations. By contrast, the Archaeoria macrodactyla, this was the very robust protein. You're still at 7 random mutations, and 90% of the genotypes, even with 7 mutations, you can explain without any interactions of those mutations whatsoever. Right? So this is weird. So essentially what I'm saying is the following. In this picture here, I'm saying that some peaks are sharp, meaning that as you start, kind of like Mont Blanc in the Alps, right? It's a very sharp peak. You start moving away from it a little bit and you fall off very quickly. So this is the yellow guy and the two ones on the bottom. And some fitness peaks are kind of like Kilimanjaro or like the Fuji mountain, right? So you go on top, and then on top, there's actually relatively big flat area that you can explore. So the shape of the mountains are different. Here, I'm also talking about different thing. The complexity of interactions of mutations is also different on different mountains. So on some of these mountains, in some of the proteins, you require a lot of quick interactions to be able to describe the genotype to phenotype connection. On some other mountains, you actually don't need those interactions at all. And again, I'm not really talking about different protein functions. These are proteins that do exactly the same thing. And we assay them in exactly the same way, right? Whether it's, the protein does the same thing, but depending on where it is in genotype space, this fitness peak has different
Quickly interrupt right now. So we still have from 5 to 10 minutes. So we probably sacrifice question time, and then people maybe can approach you. So you still have time to finish the wrap up a little bit.
Yeah. Sorry about this. I will finish this quite quickly, and then we can. I kind of need 5 minutes, and then we can go into questions. I will also come back to do a workshop, so you can ask me questions during the workshop. I think in 2 days, right?
Yes, correct.
So now that we know that we have those different GFPs, we decided, well, okay, the entire point of this exercise was not to actually just learn about specific peaks, but was to learn about the entirety of the space. Can we use the data that we assembled to start learning about points that we have not explored? So here, these small circles is what we explored, and these guys would be the guys that we predict based on the data that we have. We use I'm not going to get into this. We use machine learning to do this. And this machine learning is actually quite good in being able to predict the data that we have. And then we use these machine learning algorithms to then start predicting genotypes that are farther than 10 mutations away. And first, we took the very robust protein, right? We took the protein that can tolerate a lot of mutations. And these black circles here are the genotypes that we predicted using our machine learning algorithm that has never been seen either by evolution or by us in the experiment. And we were super, super excited about this, right? Because we can predict mutations with 12 mutations, 80 mutations, even some with 30 mutations away. And remember that I told you in the beginning, still to this day, for most proteins, we cannot even predict the effect of 2 or 3 mutations. And here, all of a sudden, we're predicting 30 mutations away. I mean, it was a fantastic result. And then we decided, well, okay, if a robust protein can tolerate many mutations, well, let's try with different proteins. So this is also a robust protein. And we were super excited to see this guy with 48 mutations away. This is phenomenal. It's like 20% divergence that we managed to completely cold turkey without any experimental poking around computationally to predict it. And then we took a fragile protein. This is a protein that is very easy to break. So we thought, okay, it's a very easy to break protein. So it would be very difficult to stick other mutations into it and have it to be functional. And it turned out to be completely not true. We were fantastically successful in predicting fluorescent protein using data of this very, very fragile protein. So for a point of with 48 mutations, half of our predictions were correct. So half of our predictions actually have a fluorescent protein that you can observe. For homework, tell me at some point, in 2 days when I return, why were we successful in predicting on a fragile protein, but we're not so successful in predicting a robust protein? It's not a simple question. We figured out only after we got this data. We thought that it would be totally the other way around. So we were actually, blah, blah, blah. We're actually quite good. And this is a final point that I want to make. That remember I told you that we wanted to understand the entirety of that space. And when I was showing you these predictions here, we're not using all of the data. We're using actually local data. So so on the top, we're only using data from Equioria macrodactyl for one GFP to start moving away from that GFP. The same thing on this bottom Clitia gregaria. We are only using the local data and they're moving away from that data, not using all of the 4 proteins, using only one of the proteins. Well, what happens when you start putting all of this data together? Maybe you can learn something about the entirety of that space. Well, unsurprisingly, so if you use AV-GFP to predict AV-GFP, you do very well. If you do AV-GFP to predict other proteins, you don't do so well, right? So, using one peak to predict another peak doesn't work so well. What about putting all of it together? This was the most surprising thing, and this is still where the field is, and this is the next questions that not just us, but everybody in the field will want to try to understand. So here's how to read this data. Here we're trying to predict one particular GFP, the Equioria macrodactyla. This here on the y-axis is how well we can predict the genotypes. So using only Equioria macrodactyla, we can do quite well in predicting that data. The real surprise is that if we start adding fitness peaks to it, right? So here, for example, the second line here, we're using Equioria macrodactyla and adding to it Equioria victoria, which is the closest protein to it, right? These two proteins are just 20% divergence apart. And by adding this additional data, you're actually not improving the predictions, you're making them worse. So that means that when we're looking at the fitness peak of those two proteins that are just 20% divergence apart, whatever other rules that determine epistatic interactions in this protein are very different rules from of what determines epistatic interactions in the neighboring protein. So by adding this data together, you're not helping to learn those interactions. You actually are adding noise because the rules by which this protein is held together have changed in 20% divergence. This is very different from what protein structure biologists will tell you. And this still means that actually we don't really understand why the rules are different between point A and point B and what we can do to actually improve on predictions and to describe the entirety of the sequence space. And what we're trying to do now is that, these 2 points is what I show you, like adding this peak to that peak doesn't help. So what we're trying to do now is we're trying to sample much more broadly between those two fitness peaks, because obviously, we can predict one fitness peak, adding the second one hurts, but maybe something in between those two peaks will actually carry this information that will tell us why is one of those peaks sharp, the other one is flat, and how do rules change between going from one place to the other. A lot of people to thank. Luisa has done all of the work on this, and then Karen was the guy who initially came up with the one of the two people came up with the idea to use the GFP and set up the experimental system, and this was mostly done with ERC and HHMI funding back when I was still studying in Barcelona and then in Austria. Any questions?
Thank you for the great talk. I believe we could have one question, but we are already very close to have to wrap up the first session. So in the audience, maybe?
In the audience, do we have some questions? Yes, we have one question.
Perfect. That would be the question. And then everyone else later contact directly or at the workshop.
Yes, thank you for the lecture. And could you please show us the first slide where you introduced this landscape, or even we can ask. Yeah.
This one.
No, after this, with a landscape map. Yeah, here. And I understand that it probably depends on the purpose of the study and how many genes were analyzed, but I want to ask what are the usual size of landscape maps like this, and
Oh, fantastic question. So GFP remains one of the most studied proteins. We have at this point, let's say 120,000 genotypes that we've surveyed. There are some smaller pieces like a promoter has been looked at. There's maybe a few 100,000 genotypes that we looked at. There is a particular protein where the active site has been looked at in more divergence. But this is a really good question, because at this point we still don't know how much data is enough. And here's the problem: the volume of data of GFP is 20 to the power of 250. So this is more than the number of all quarks in the universe, right? So 100,000 is smaller than one electron in the universe. How many, would 3 electrons be enough to understand the entirety of the field? I don't know. And this is a very good and very open question. We have no idea how much data we need.
And what about the units of these plus and minus sign regions? How do you measure the fitness exactly? I mean, in practical unit.
It's very difficult to. This is actually one of the things that is actually important. So people are doing these fitness landscapes of different proteins. For GFP, this is just some measure of the level of fluorescence, and we're taking a logarithm of that. So a lot of other people, when they're mutating genes, they would be looking at the rate of growth of bacteria. And then you can also take, the logarithm of the rate of growth in, let's say, stationary or in an exponential growth phase, and that would be your measure of phenotype. The question is, how would you then compare those two landscapes? How do you compare level of fluorescence to rate of growth? It's totally open question. We don't know. So really, just I think you're really asking fantastic questions, because for the last 100 years, since Sewell Wright has created these landscapes, what we lack is data. We don't lack theories. There are tons of theories. There has been, 1 million papers written on the mathematics of what happens to the fitness landscapes, of how you go from high dimensionality to low dimensionality, of what happens to the rate of change of genotypes. We still lack data. We lack consistency of data. We lack data that are collected in the same environment, and we lack volume of data. We need millions and millions of these things measured in exactly the same way, where the phenotype is measured in exactly the same way as well.
Yes, thank you. And one very quick question from previous slide. Can you please show us the graph?
Yep.
Before this. Yeah, and as I understand, this, for example, CT and TC will be highly represented in the population, like compared to compared to CC, for example, or something like this? Or from the right, as I understand, there will be not a lot of organisms with AA genotype, like.
Yeah. So populations will not be able to occupy areas of low fitness. So these black circles will be completely empty. And to what extent the population will spread on green genotypes will depend on two factors. One, whether or not there's differences in fitness between those green genotypes. Here I'm assuming that they're equal, but that's not necessarily the case. And two is the size of the population, so by genetic drift. Because there is effectively an infinite number of these green spaces, if you have a very small population, that population will occupy a very small space. So that is also kind of, where population genetics intersects this field, I would say.
And so will they reach some kind of...
Sorry, we have to finish now. Sorry, please continue with questions. This is great, but we have to move on to next speaker. Yes. Sorry.
Sorry to be late. Please email me. I'm Googleable. Just email me if you have any questions, and I'll see you in a couple of days.
Thank you very much. Apologize for confusion, and let's move on to next speaker. Thank you.
Thank you.
Okay, we will start introducing now, I think, and while things being prepared, I think presentation is not shared yet with online audience. So it is my pleasure to introduce Svetlana Serga. She started her higher education in Kharkiv National University. Then she received PhD from Taras Shevchenko National University of Kiev on Drosophila bulbaceae interactions. And now she is a scientist in INRAO and CBGP in Montpellier, in France, and is a receiver of Marie Curie, very prestigious award. So the floor is yours. Welcome.
Thank you very much. I'm happy to be here. And first, I want to tell you my story about this hall, because for me, it's second time I will present my study at the Uzhgorod National University. First time, it was 15 years ago when I was master student. And now I'm really happy to be at the same place and to present development of my research and also to give some background in the topic. So I want to start with words of Lynn Margulis, that life did not take over the world by combat, but by networking. So Lynn Margulis was a scientist who proposed endosymbiotic theory of eukaryotes origin. And I think her words now are very important and actual for current understanding of interactions, the role of interactions between organisms. And here on slide, you can see one of the ancient presented examples of symbiotic relationship, which was described by Herodot. It's Nile crocodile and bird who help Nile crocodile with teeth. And this bird can take some pieces of food from Nile crocodile's teeth and help to survive for Nile crocodile this way. And from the side of Nile crocodile, this interaction is very successful too, because he doesn't have problems with his teeth. And people know a long time ago examples of such interactions. And now we call such interactions symbiosis. This means coexistence of taxonomically different organisms. And this term was proposed by Heinrich van de Bary. And now we know many examples of such interactions, and most of interactions between organisms of different taxons we can describe as symbiosis. Debarry worked with lichens, and lichens are very good example of symbionts. It's symbiont of algae and mushrooms. Also, there is another great example of symbiosis. It's squid and bacteria, which are in its special organ, luminescent organ, and only because of existence of such bacteria in the organ, squid can have light and can survive in dark environment in very deep environment in the sea. The other great example of symbiosis is gut-human microbiota. And I think all of you know that we have microbiota inside us, and this microbiota have great influence of our health and on our survival too. Also, coral reefs are another example of symbiosis. And in plants, we have azote fixating bacteria interaction with plants, which take possible fixation of nitrogen. And clownfish interact with sea anemone. In this case, sea anemone can eat different organisms, but not clownfish. And at the same time, clownfish is living near anemone, and because of this fish, other fish can be closer to anemone, and anemone can kill them. So it's I gave you some examples of very beneficial interactions between organisms of different species, but symbiosis is more general term, and we understand all interactions which can be between organisms of different taxons. And we can divide such interactions into different groups. First, neutralism. Neutralism means when species are living near each other, but they don't have any interactions. They are not beneficial and they are not harmful. And here you have example, in the forest, we have different trees and they are just living near each other and there is no interaction between them. Also, the other example is an antagonism when such interactions are harmful for both sides, and great example is resistance of different bacteria to each other, and you can see in the slide like different bacteria can grow only separately. And we have many examples in the world of such interactions. Next part is amensalism. Amensalism means when it is neutral interaction for one organism and harmful for another. And you can see one tree can grow up, but it influences trees which are living close to this one tree. But the most interesting interactions are more close interactions between species, and they can be different types. One is more ecological, it's predators, when one species can eat another. and we have more close interactions, one of which is parasitism, when this interaction is harmful for one species and very successful for another. And as example, we can see tick, which can parasite in humans, for example, and this interaction is good for ticks, but not good for humans. And the other interaction is commensalism, when this interaction is very beneficial for one species, like in case of orchids, and is neutral for the other species. So orchids can live in the branches of trees, but they don't have any harmful influence on the trees. And the most close interaction is symbiosis or mutualism, when this interaction is beneficial for all species included in such interaction. And we have different types of symbiosis depending on influence of organism on each other. We have facultative and obligatory symbiosis. In case of facultative symbiosis, species can live without each other, but in case of obligatory symbiosis, when we remove one species, the other will die. And in all such types of interactions, we have interaction between species, and the main question is how this interaction can evolve and which factors can influence such interactions. And we have several hypotheses about this. And the main thing is that parasitic and mutualistic interactions are very different, and mechanisms of interactions can be different between host organism and symbiont. There are some models which can describe evolution of interactions between host and symbiont or host and parasite. And we'll start with parasite. We have some evolutionary genetics models which can describe such interaction and coevolution between two species. And you can see in the slide graph with genotypes, frequency, and generations. In general case, theories predict that for parasitic interactions could be genes inside parasite which have some allele frequencies, and could be genes inside host with some allele frequencies. And in host and products of such genes can recognize each other, and we have variation inside host genes, we have a variation inside parasite genes, and when host genes and parasite genes match with each other, they recognize each other, and it could be infection in this case. But when there is no match, there is no infection. So parasite evolve to be matched with host, and host evolve to escape parasite. And in this case, we have fluctuating selection, and we have waves of dynamics of frequencies in genotypes of host and in genotype of symbiont of parasite. In case of mutualism, we have another case. For example, it's the same graph with genotype, frequency, and generation, number of generations. And we have two hosts, host A and host B, and symbiont. In case of beneficial interactions, when symbiont can give some fitness benefits to the host, we will see increase of genotype frequency of host and symbiont. If it's obligatory symbiosis after some generation, it could be fixation. So we will have 100% of infected individuals in the populations with a certain host genotype. But if symbiote is not beneficial, we will have reduction in frequency and decrease of symbiont in the population. And for now, I started to think about another question from where we can have symbiont in this first point. So all such predictions started from the point that we have symbiont and parasite and host. But from where could be symbiont in the host? And most theories predict that symbiont acquired from free living bacteria, and then they started to be parasitic, and then they evolved to mutualistic symbionts and started to be obligatory symbionts. We have many examples of symbionts, but now I want to stop on one group of symbionts, which called reproductive parasites. They are bacteria which can influence reproduction of their host. There are many bacteria which can influence reproduction of host, mainly in invertebrates. Among them are Spiroplasma, Cardinium, Rickettsia, Wolbachia, and I can say about Rickettsia too, and more and more species we discover who can influence reproduction of the host. When I told about influence of reproduction, I mean few reproduction manipulations which can induce such bacteria in different organisms. First, it can be feminization. In this case, genetically genetical males can develop as females. And at the end, in the population, we have only females. It can be also parthenogenesis induction and a special type of parthenogenesis when females can give only female progeny. The other manipulation is male killing. In this case, infected females can give only female progeny or mainly female progeny, because it could be some males in the progeny, but mostly females. And as you see, in all such interactions, we saw change of sex ratio forward more females in the progeny. And the fourth manipulation is cytoplasmic incompatibility. In this case, in crosses of uninfected female with infected males, we don't have progeny. Maybe you can have question why in all such interactions bacteria can influence sex ratio and why in this case in populations would be more females than males. Such bacteria can transmit only or mainly maternally. It means that they have vertical transmission, and infected females can give in next generation infective progeny, infected females and males. But there is no or very rare horizontal transmission from inside one generation. And also, in this case, males can't transmit the bacteria to the other generation. So we have very special system when only females can transmit bacteria. And success of bacteria really depends on the number of infected females in the population. And general model is that infected individual can migrate or can move to the population. And in this case, we will have infected population, possibly, but how it happened and which factors can influence this and how such interactions evolve is the main question. And such processes were studied mostly in endosymbiotic bacteria Wolbachia, which is one of the most prevalent bacteria in the world. It can infect more than 50% of all arthropod species, and also it can infect nematodes. Wolbachia can be obligatory symbiont or reproductive parasite. And it's maternally transmitted bacteria, as I described. And Wolbachia can influence fitness of infected individuals. It can infect many species like Drosophila, nematodes, isopods, ticks, different species of mosquitoes. And formally, Wolbachia, all Wolbachia bacteria classified into one species, Wolbachia pipientis, but such species can divide it into several supergroups. And with time, we have more and more supergroups studied because people started to work with more arthropods and found bacteria and found Wolbachia, and then we can add more and more supergroups because of this. But formally, there is only one species of Wolbachia. The most prevalent are A and B supergroups, and such bacteria can infect arthropods, and usually they are reproductive parasites. The other prevalent supergroups are C and D, and they infect nematodes mainly, and such bacteria are obligatory symbionts of nematodes. So Wolbachia is a very good system to study different types of symbiosis, because in one species it can induce reproductive manipulation, it can be parasitic. In other species it can be symbiotic or even obligatory symbiotic. And some examples of Wolbachia and how such bacteria can influence their host. And the main thing in this case that effect of Wolbachia on the host depends on type of Wolbachia or of Wolbachia strain, and host genetics also influence such interactions. So we have two components. We have type of bacteria and type of host. And Wolbachia can induce male killing phenotype in some species of Drosophila. In this case, infected individuals can give only female progeny. And it was counted that infection level of such bacterian population is near 30%. And it is 5% of selective advantage of infected individuals of such in such species. And also such phenotype can influence sex ratio, and in this case it would be more females than males. Also, Wolbachia can induce parthenogenesis in species with facultative parthenogenesis. And for example, in Coleoptera, In this case, Wolbachia can infect some populations of host species, and in such populations, such populations can be parthenogenetic, but from the other side, there are some populations which are not parthenogenetic and contain individuals of both species. And Wolbachia can induce feminizations in isopod and butterflies. In this case, genetically males, genetical males develop as females, and sex ratio is in sex ratio we have more females than males. And the most interesting phenotype for me is cytoplasmic incompatibility, which was described first phenotype of Wolbachia in Kulik's pipiens. And because of such host, Wolbachia was called Wolbachia pipientis. And in this case, we can imagine 4 types of crosses of infected and uninfected individuals. And infected individuals can cross with both males infected and non-infected and give progeny. But in case of uninfected females, in cross with infected males, they don't have progeny. And it gives fitness disadvantage for uninfected individuals. But in Kulik's, the system is even more complex because there are not one Wolbachia strain in this complex. And if individuals are infected with different Wolbachia strains, it could be the same, no progeny. So in this case, we have like incompatibility system, which induced by bacteria. And to study such processes, we have great model. It's Drosophila, and we have different types of interactions of Wolbachia with different Drosophila species. And one species is Drosophila simulans. And it can be infected with WRI strain of Wolbachia. And I told you that effect of Wolbachia depends on Wolbachia strain and host species. And so this strain can induce very high levels of cytoplasmic incompatibility in Drosophila simulans, and means that in such incompatible cross, we have near 100 reduction of progeny in Drosophila. The other species is Drosophila melanogaster, and it is infected by W-male strain of Wolbachia. This strain doesn't induce high levels of cytoplasmic incompatibility. And third species is Drosophila Suzuki, is agricultural pest species, and it's infected by W-Sus strain of Wolbachia. As you remember, now we want to understand processes which can be inside populations of host species. And such model is very good to describe such products. So Wolbachia population dynamics of species in which Wolbachia can induce cytoplasmic incompatibility can be described by 3 parameters. First parameter is maternal transmission. Maternal transmission means a number of infected individuals of the progeny of infected female. So as Wolbachia can transmit vertically, it means that some percent of progeny would be infected, And this parameter is describes this person. So it can be 100, it can be 90, it can be 50 or something like this. The other parameter is reduction in relative hatch rate. It means cytoplasmic incompatibility. If we have high cytoplasmic incompatibility, we have great reduction. If low, we have low reduction. And third parameter is a reduction in relative fitness. It means that Wolbachia can influence not only reproduction, it can influence also fitness parameters of the host. Using such parameters, we can model Wolbachia infection frequency in the population. And we can count parameter p hat, which means level of which means level in after which we can have difference, we have dynamics in infection frequency, because dynamics of infection frequency is frequency dependent. And so p hat is parameter which can describe this, and it's depend on level of reduction of CI, of cytoplasmic incompatibility, and fitness reduction. In simple case, it's fecundity. Level of p hat can be different, and after when infection frequency of Wolbachia or number of infected individuals in the population reach such level, infection frequency will decrease in the population. And what we can infect? This is cytoplasmic incompatibility. I already described you, but it's in Drosophila. Now some explanation about fitness. Fitness is we can measure fitness different ways, and we can do this in Drosophila different ways too. We have many approaches to do this, but the main thing is that to measure fitness is very difficult to have a good approximation of fitness. But we can measure some parameters, and we can have some idea in Drosophila about such parameter for modeling in populations. And also, hypothetically, we can understand that symbiont impact host fitness different ways. So if it's parasitic interaction, symbiont will decrease fitness. If interactions are mutualistic, it will increase fitness, and also could be neutral interactions when we don't have any influence of symbiont to the host fitness. And also in this case, we have different influence of selection. And I really like this picture because it's really describe what we can have.
Да. Мгм. Сейчас я тогда найду. А так, в целом, вроде нормально. Я не вижу ничего такого. Так я просмотрел еще раз видимо строчка.
Хтось.
Падало ці вас чути.
And this is real data from populations
Мгм.
in Egypt, Australia. In this mosquito, people introduce Wolbachia of strain from Drosophila, and they have a very good data set of temporary data about infection frequencies in such population. And as you can see in the slide, there is great variation in infection frequency, and it can be explained by drift, for example. But in one case, when infection frequency was near 50%, we have a great increase in infection frequency, and after this, it reached fixation. And in such system, we have Wolbachia, which can induce cytoplasmic incompatibility. This picture means that in population before basal level, we can have some influence of drift on infection frequency and less influence of cytoplasmic incompatibility or no influence of this. But when infection frequency reach some level, we have a great influence of cytoplasmic incompatibility and infection frequency fixed. Influence of reproduction parasitic phenotype is frequency dependent. And what we have really in nature in Drosophila, great example of influence of cytoplasmic incompatibility on Drosophila is WRI infection in Drosophila simulans, which was described in Australia. And I will remind you that this strain have high levels of cytoplasmic incompatibility. Also, it has negative or neutral fitness depending on the paper. And it was described perfect maternal transmission in this case. In Australia, there is big group working in Wolbachia, in Drosophila, and they found very interesting situation. When they started their study, they found very little level of infection of one strain of Wolbachia, but then in 2 time points, you can see my. Yes, and in 2 populations, they found another strain of Wolbachia, which called WRI, and then it really quickly spread in all populations that they tested. And this strain is can induce cytoplasmic incompatibility. It means that in this case, we have low p-hat and infection frequency can increase greatly because of cytoplasmic incompatibility. We studied Wolbachia and Drosophila simulans in Odessa population in Ukraine, and we find some variation in infection frequency. Here we have different infection frequencies, and we found fluctuation, possibly like because of drift, and but levels of infection were very high all time when we investigated this, and it's in the line with prediction of CI influence on this. In Drosophila melanogaster, we have a very different picture because of Wolbachia and Drosophila melanogaster can induce only low conditional cytoplasmic incompatibility, but it has positive effect on fitness and high maternal.
Czy będzie potem wracał tym samochodem tutaj? Bo ja wzięłam na wszelki wypadek laptopa, bo będę raczej mówić tą prezentację z stacjonarnego, ale tak na wszelki wypadek. Tak, i o 16 to kończę, więc nie będziesz wychodził przed 16, nie?
Дуже вибачаємося. Завідьте себе.
Na zupę się umówimy.
We have great variation.
O 7.
And in this case, we think we have.
To może być trochę mało.
Excuse me, please, everyone, check if you are muted. There is someone talking in the background. Sorry, please.
But very rare we have fixation of.
Możesz wyjechać, co pół.
And the other example, which I will describe more in details, is Wolbachia in Drosophila Suzuki. Drosophila Suzuki is spotted twin Drosophila, and it's invasive agricultural pest species. It can infest many species of berry or cherry, strawberry, and other, and it induces loss of production of such things in different countries, and now it's really big problem in Europe and United States. Drosophila suzuki is invasive species. It was originated in East Asia, and it invaded worldwide population very recently. It was found in 2008 in Europe and United States, and then it moved to South America and Madagascar, and now we have more and more evidence about Drosophila suzuki in Africa. But what about endosymbiont infection frequencies in case of invasive species? It could be very different story than in previous examples which I gave you. So we have host population, like native population, which can have different alleles or different variants of host and endosymbiont too. And then during invasion, usually we have reduction of genetic diversity of the host, and we usually predict the same for symbiont. And also it can be reduction of infection frequency of symbiont, or it could be 100 infection frequency by chance, just by chance. We investigated Wolbachia prevalence in Drosophila suzuki populations worldwide, and we find rather high infection frequency in native populations in China and Japan. We tested more populations from Europe, and we found mean 35% of infected females or males in the population in Europe, and we have great variation of infection frequencies in European populations. But infection frequency of Wolbachia in populations from Americas is very low. It's near 13%. And the main question, why we have such situation, how we can explain this? First question, do we have cytoplasmic incompatibility in Drosophila suzuki, which is one of the parameters which can describe dynamics of Wolbachia in host population? And there were two papers with not big sample size, in which were tested cytoplasmic incompatibility in Drosophila suzuki from Europe and from United States, and they didn't find cytoplasmic incompatibility in such flies. In Europe, they found some level of hatch rate reduction in population from France, but it was not statistically significant according after's measurement. And we wanted to understand really, do we have cytoplasmic incompatibility in Drosophila suzuki? To do this, we made screening of all live strains of Drosophila suzuki which can find, and we found 49 strains and in labs in Europe, and we tested them by PCR, and we found 13 Wolbachia infected strains among them. They were from different parts of the world, from Japan, China, Korea, from Europe, from France, and from Italy, and also we had strains from United States. We sequenced genomes of such strains by Illumina, and we wanted to understand, do we have the same Wolbachia in some strains, or do we have different Wolbachia? And we found that there are different that genetic diversity, genetic diversity of Wolbachia is not very high in Drosophila suzuki, but we have some variation. According to such variation, strains can be divided into some groups, but such variation is not very big. It can't really explain big difference in phenotypes, or we can consider them like different strains of Wolbachia. And also strains from Europe were very close, and only one strain was very different. And then we made a really crazy experiment to measure cytoplasmic incompatibility. We made for 13 strains, we made 4 types of crosses, all possible crosses. And in all such types of crosses, we tested 20 females. And for each female, we analyzed 5 petri dishes with eggs and larvae. And in total, we analyzed more than, I think, 5,000 of petri dishes to have such results. And then we measured cytoplasmic incompatibility by the ratio of hatch rate. It means ratio of larvae of eggs and hatched from them larvae, we compared this in incompatible and compatible crosses. And what we found in populations from Europe, you can see here in the slide 4 types of crosses. One type is incompatible cross, and you can see like the same color here and here, and 3 control crosses. And we found reduction in hatch rate in strain from Trento, and this reduction was significant. So we found cytoplasmic incompatibility in Drosophila suzuki. It was cool, but it was very similar to results which was in the paper described French populations. But what we found great, we found in one strain, we found very high reduction in hatch rate in strain from France, Balandran. And in this case, we had 70% of reduction of hatch rate, and it was really unusual. And in 2 other strains from Europe, we didn't find cytoplasmic incompatibility. So we had great variation in phenotype in European populations, but we don't have really difference in Wolbachia genetics in such strains. Also, we tested strains from America in invasive populations. European populations are invasive too, and we didn't find consistent results. We found in 2 strains, we found reduction of hatch rate in control cross, in test cross, sorry, where we expect to have reduction for cytoplasmic incompatibility, but we had the same reduction in control cross with the same males. So it can be cytoplasmic incompatibility, or it can be effect of male genetic effect, like this. And we didn't find cytoplasmic incompatibility or reduction of hatch rate in 2 other strains.
We have about 10 minutes, I think, a bit less maybe, and there won't be already time for questions. So,
Area.
but yeah.
We find a similar picture with the reduction of hatch rate in population from China, but also it was the same. We found effect of male genetics on this parameter. We found cytoplasmic incompatibility in European populations, but not in Asian populations. But cytoplasmic incompatibility alone can't explain difference in infection levels in populations of Drosophila suzuki. And in our data set, we also tested fecundity. It can be measure of fitness. And we measured number of laid eggs of Drosophila into crosses with Wolbachia and without Wolbachia. And we didn't find Wolbachia effect on fitness in this case. So Wolbachia is not don't have deleterious effects in the laboratory to fitness of Drosophila suzuki. So we really can't explain by fitness disadvantage low infection rates in invasive populations of Drosophila suzuki. And according to this model, what we predict that in native populations, we have higher p-hat and we have like this situation. And in invasive populations, we have this situation when infection frequency tend to decrease. But to have clear understanding of such system, we need to do more investigation. And I want to acknowledge many people who was involved in work which I presented. I want to say thank you funding which I had from PAUS and INR program in France, and also Maria Curie for Ukraine. And part of work was done under this project. I want to say thank you, Nicolas Rod, with whom I'm working at CBGP in France. He gave me great advice in my job, and we made really good project with him. And also people who with whom I'm working at CPGP, Matthew Gauthier, Arnaud Stoup, and students who helped with such a great and big experiment with cytoplasmic incompatibility, Chulin, Montesiera, and Marie. And also I want to say thank you, my former supervisor, Irina Kozeretska, and people from this lab. It was great time what we spent. Alexander Mastrenko, with whom we made many interesting investigation in Drosophila melanogaster, and Anton Lavrimenko, our former master student who started this project in Drosophila suzuki, and Venera Tukmaeva, she will have a workshop today with Andrea Betancourt. She also involved in this work. And Emily Knott from Finland, she involved in work to test infection frequencies of Wolbachia and Suzuki, and also technicians from CBGP, Candice, Anne, and Lily, and people who gave flies, and all of you for your attention. Thank you.
Thank you, Svetlana. So now we have time for 1 or 2 questions, hopefully not too long, so we can move to the coffee break, and there are still a couple of announcements before that. So please, audience first, in-person audience first.
Yes. Can you inform us how incompatibility works? What is the mechanism of incompatibility?
Yes, thank you. It's a great question. I think I have even slide for this. Yes. So for incompatibility, short question, we don't know exactly what happened, but we have some models about this. There are two models, very competitive models. One was originated in mosquitoes, and it's called toxin antidote model of cytoplasmic incompatibility. In this case, we don't have Wolbachia in the sperm because it's too big to be inside sperm, but it's we have some toxin proteins inside sperm, and such sperm can infect uninfected females and infected females, like not females but oocytes. In case of uninfected oocyte, we have some cytological defects in genetic material, and in this case we don't have development of embryo. In case of infected oocyte, we have Wolbachia inside, and we have proteins which called antidote inside oocyte, and such proteins can interact with toxins and rescue embryos. But it's in theory. The other model is host modification model. It was developed in Drosophila. Okay. Yes. In this case, the same. We don't have Wolbachia in the sperm, but there is a modification of DNA in the sperm, like here, which occur pre-fertilization. And then modified sperm can infect uninfected oocyte, and we have death. And if it infect infected, it made with infected oocyte, we don't have death, we have rescue. And according to this model, like last paper last year on 2024, they found that Wolbachia induced DNA damage in such DNA damage of such oocytes, and because of this we have death of zygote, but this model really can't explain rescue phenotype. How it works, we don't know.
Thank you. I don't think we have more time for questions, but Svetlana is co-organizer. She's present through entire workshop, and then please ask questions, discuss in person. So several points. I think you'll need to change the room for next speaker. And now it's
for this.
Sorry.
Yeah. I will give this information, Alexander.
Yeah. Oh, okay. And there is another announcement. So Natalia sent everyone a new program which has a different link to today's workshop on from Andrea on abstract analyzing abstract. So please, those who want to join or who subscribed already to join it, or you have been assigned to that workshop, please check the email and you will find the new updated link. So from my side, I think that's it. Question to everyone: can online audience stay in the room, and then you will on the spot in Uzhgorod you will rejoin from a different auditorium? How this will work?
Yeah. Okay. Thank you. I have this information.
Perfect. Okay.
Yeah. Did you finish?
Yes. Yeah.
Okay. Thank you. So, information. What would be next? Next, we will go for the next lecture to the biology faculty of the university, and we will go all together. We will have coffee at the biology faculty, and we will have lecture in half an hour. Then we will spend all time today at biology faculty. We will have all lecturers in the other building. But students who will attend in Walter workshop in bioinformatics, basics of bioinformatics, will do this workshop in this building. So you after lunch, you will come back to this building and will have workshop there. All other students will have all workshops in biology faculty, and you should have a list of participants for each workshop, and you can find your name and go to the workshop in which list you are. Okay?
This workshop. Please hand up who seen it for Walter's workshop for on bioinformatics. 1, 2, 4. So around 10, yeah. Short information. This workshop will be not their auditorium. Please wait for me when we finish, and I show your auditorium for this bioinformatics workshop. Okay. When do you back from biological faculty? Okay. Thank you.
Hey, still managed to join, all good. We are waiting for in-person audience to get online, back online.
Yeah, right. Okay. Hello there.
Hello, hello, yeah.
Just checking you can, okay, got no video.
Yeah, I think you should be allowed to turn on your video. So, oh yeah,
There we go.
perfect. Yes. Hello.
How are you?
Good. How are you?
I'm well, thanks. Yeah. Yeah.
Yeah. So well, yeah. There was need to change auditorium from each in-person audience will be participating, so that might take a couple of minutes still.
Yeah, yeah. That's no problem. Don't worry.
In the meanwhile, you maybe can try to put the presentation on if everything
Yeah,
works.
let me have a go.
Also, yeah.
So you should be able to see the slides now.
Yeah, it's perfect. Yeah.
Well, good. Good.
Yeah, thank you. Yeah. Yeah, so to briefly recap, so it's 45 minutes for the talk itself and 15 minutes questions, discussion. So we sometimes had to readjust a little bit. If you don't mind, we might share like an email or maybe some other way how to contact you if people have more questions. Because last couple of speakers, we just had so many questions, then yeah, it became impossible. 15
Sure.
minutes seemed to be too little time. Yeah.
Yeah, well, I don't even know how long my talk's going to be. I wrote it last night.
That's
So
how we do it.
In the proper professorial way, I'll either finish 15 minutes early or 15 minutes late.
That's no
Yeah,
problem.
no problem sharing my email address with people. That's fine.
Okay, sounds good. Yeah. Um, Taking a bit longer. Sorry.
Yeah, no problem.
So it should be now.
Okay.
It should be soon, yeah.
Okay.
Yeah, there was so actually the reason why it's taking longer also there was airstrike alert apparently, so it's a little bit, yeah, okay. Yeah, at least they're in the room already, so yeah. Okay, Natalia is in, so I guess we are already close to start.
If somebody turn to our biological faculty Wi-Fi, that because signal is not high, and we can. Будь ласка, якщо ви підключалися до Wi-Fi логічного, відключіться на момент лекції, тому що щось трошки провисає сигнал. Розпочнемо.
Start already?
Okay. Am I good? Am I good? Am I good to start?
Yeah, I will say one just word. Yeah, so if there are if no one texts me that they cannot hear us, then everything is fine. It looks like everything is fine. So yeah, hello, welcome everyone to the third speaker of today, Professor Robert Nell from University of Hull. He specializes in evolutionary ecology, adaptation to changing environment, sexual selection, mating systems, animal weaponry, and several other topics. So please, you're welcome. Thank you for joining to give a talk at Ukrainian School for Evolutionary Biology.
Okay, thank you very much. I hope you can all hear me, and I apologize for speaking entirely in English and not in Ukrainian. Greetings from the north of England, where the weather is very north of England at the moment. We've got grey skies and rain, and it's cold. Anyway, so I'm going to be talking about the evolution of animal weaponry. Just to give you the structure of the talk, we'll start off by talking about animal weapons for interspecific interactions, but those are quite boring, so we're not going to talk about them for long. And then we're going to get on to the more interesting subject of animal weapons for intraspecific interactions. And there's a number of subtopics that I'm quickly going to talk through. When should males, and it's usually males, compete aggressively? When should they not? When do we get lethal contests between individuals of the same species? The normal situation is that we don't, but sometimes we do. I'm going to talk about alternative mating tactics, where you get usually males again in a species adopting different tactics and often carrying different kinds of weaponry. And then the last thing we're going to talk about is why weapons that are used in non-lethal contests between animals are so incredibly diverse. This is my eclectic take on what I think is on what I think are interesting questions in this subject. I should say now this is a much, much bigger subject than I will be able to cover in 40 minutes or 45 minutes. And there's a lot of things that I'm just going to ignore. So please excuse me for that. We're only going to cover what I think are some interesting questions that we can talk about in a fairly brief way. So let's kick off with the kind of weapons that evolve for interactions between species. And what we're talking about here are the kind of weapons that are used either for predation or for defense against predators. So this lion here is obviously showing off her teeth, and those are weapons, or at least some of them are weapons that she uses during interactions with her prey, and she uses these weapons to subdue and kill her prey. And then the other teeth she uses to turn the prey into more lions. Some of these weapons for interspecific interactions are pretty spectacular. This is an antlion larva here, and you can see it's got these enormous mandibles that it uses to catch ants. Very similar, to catch ants that fall into its trap. So some of these weapons that animals evolve for interspecific interactions get large and quite spectacular. Here's another example. This is a mantid, and obviously we have what are called the raptorial forelimbs of the mantid that it uses to catch its prey. So these are quite impressive weapons again. But most weapons that are used in predation tend to be reasonably small and very functional and really quite boring. So if you think about predatory mammals, they've all got pointy teeth and sharp claws, or most of them have got sharp claws, but they don't have any huge teeth or any diverse branching teeth. There's not a lot of difference in the form of the teeth between them. And the same thing, if you look at the mantids, we're talking about, half of an order of insects here, and their raptorial forelegs are all pretty much the same. And when we're thinking about weapons that are used in predation, there's no really hard questions there that I can think of from an evolutionary perspective. Individuals that have weapons that are the right size and have the right amount of pointy stuff on, they tend to get lots of things to eat. They do well, they make lots of babies, and so those weapons are selected for. They're not hugely diverse. there's not a lot really going on in terms of interesting evolutionary biology. So I'm not going to talk much anymore about weapons for interspecific interactions. Instead, I'm going to focus most of this talk on the evolution of weapons that are used in interactions within species. So when we're talking about weapons that are used for intraspecific interactions, we're thinking about weaponry that arises either via sexual selection, whereby individuals that are usually, but not always, males interact with each other in an aggressive way to try and monopolize either females for mating or resources that will then allow them to access females for mating. So we can get sexual selection, and we can also get what's called sociosexual selection, where individuals will interact with each other either for purposes of acquiring matings or for purposes of acquiring social status within a group. So a lot of the aggressive interactions that you might see within a troop of baboons or a herd of elephants, for example, you might want to class those as sociosexual selection. And we've got a nice example of an animal here where the male is carrying some fairly spectacular weaponry that's used in contests with other males in order to acquire access to females. This is a kudu and his family. You can see there's Mr. Kudu on the right-hand side of the image, and he's got these large twisty horns. And then there's two female kudu on the left, and then there's baby kudu in the middle. So this is a successful male kudu. He's got a family with two wives, and he's got a kid. And part of his ability to be successful is the fact that he's got this big set of horns that he's then able to use in interactions with other male kudu and basically chase them off and maintain his access to those females. And these weapons that we find on usually male animals for intraspecific interactions, particularly contests over access to females or access to resources that females might themselves want to access, these weapons are very, very different from the kind of weapons that we see used in predatory interactions. They're often large, they're often very costly to the individual to carry, and they're extremely diverse. So just to give you some examples of the kind of things that we tend to see, we've got traits that you might want to think of as weapons that are used in interactions between males, but that don't actually have any functional use. So this is a stalk-eyed fly. This is Teleopsis dalmani. And the males of this species have these very long eye stalks. There's female choice for these eye stalks. Female diopsids, which also have rather shorter eye stalks, female diopsids preferentially mate with males with long eye stalks, but the males also use these eye stalks in aggressive interactions with each other. So if you watch two diopsid males, you can if you take a couple of diopsid males and don't feed them for an hour or so, you can pop them in a petri dish with a little drop of food in the middle, and they'll fight over the food, and you can sit and watch it. And first of all, they'll match up their eye stalks like this. And if one of them's got much longer eye stalks than the other will back off. But then if their eye stalks are about the same length, what they'll do is you can't really see what I'm doing because of the camera, but they'll hold out their forelegs underneath the eye stalks and wave them to give a kind of signal reinforcement. And if one of them still doesn't back off, they will then actually fight. They fight kind of like little girls in the playground. They sort of do this to each other. But they're using these eye stalks as signals of what we call resource holding potential, as signals of their ability to defend that food item. So this is one end of the spectrum when we're thinking about the kind of weapons that animals carry for intraspecific interactions. We then have traits that are used as tools, as weapons in non-lethal contests. So this is a dynastid beetle in Borneo. It's The genus is Chalcosoma. I'm fairly sure it's Chalcosoma mollencampi, but it could be Chalcosoma atlas. If any of you are dynastid experts and can identify this to species, I'd be delighted to hear. But you can see that this male beetle has got 3 very large and long horns, 2 of them on its thorax and 1 of them on its head. And when these beetles have contests with each other, they use these horns to try and lift the other beetle up and chuck him off a tree. So we've gone from traits that are signals of resource holding potential, to traits that are actually used as weapons during contests. And quite often, these traits that are used as weapons are also used as signals. So before they actually fight, these dynastid beetles will assess each other on the basis of the size of their horns, and will maybe back off from a fight if the other beetle has got very large horns. And then finally, we also have weapons used in intra-specific contests between animals, between males, that are actually used in lethal contests. And interestingly, if we think back to our examples of weapons used by predators, these weapons that are used in lethal contests tend to be quite simple and very functional again. So this is a photo from James Cook at the University of Queensland, I think, and he works on fig wasps. And this is a male fig wasp here, and you can see he's got a big head and he's got a big pointy pair of mandibles. And within the fig, this male fig wasp will engage in lethal combat with other males in the hope of killing all the other males in the fig, which will then give him a monopoly over all the females in the fig. And these mandibles that this fig wasp has, these are not used as signals. They're not waving their mandibles at each other before they fight. These are simple, straightforward, and lethal weapons that they use. So that's the kind of variety of animal weaponry that we see. And there's this gradient of how these things are used, from pure signals to pure weapons. Most of them are somewhere in the middle and are used as signals and also as weapons in these contests. One interesting point that I've put in, because I like dinosaurs, is that these things have been going on for a while. And if we look at the diversity of extinct animals, we find that there's an enormous amount of diversity that appears to be associated with contests between males in much the same way as a lot of the diversity of modern animals is associated with these contests between males. And this is one of my favorite examples from a paper by an American paleontologist called Andy Farkey. And Andy Farkey, so there was a lot of debate in the early noughties, in the early 21st century, over whether things like the horns and frills of ceratopsian dinosaurs like Triceratops, or the crests that you get on hadrosaurs, were sexual signals or sociosexual signals, or whether they were something else, whether they were defensive traits to use against predators.
Sorry for interrupting. I think the room was lost from us, so they are right now trying to reconnect.
Okay.
I'm sorry.
No worries.
1 minute. I will just ask what's the status.
Yeah.
Yeah, I'm sorry.
It's fine, no problem. Oh, I think we're back.
Oh yeah, the room is back, I think. Yeah.
Okay, so when did you lose me? Had I started talking about dinosaurs?
I think the dino, yes. Around
Okay,
that time, I think, yeah.
Okay, so back to dinosaur weapons. So in the early 21st century, there was a big debate about whether things like the horns and the frills on ceratopsians like Triceratops, or the crests on hadrosaurs, were sociosexual signals, and in the case of ceratopsians, whether weapons, or whether they fulfilled some other role. And some very prominent.
Someone just joined and didn't mute themselves. Please mute yourselves.
Okay, so there was no problem. There was there was this debate over whether things like the horns and frills on Triceratops are weapons that evolved via sexual selection, or whether they were species recognition signals, for example. And some very prominent American paleontologists were very insistent that they were species recognition signals, including the guy who was the paleontology advisor on Jurassic Park, believe it or not. Those of us on the other side argued that they were clearly sociosexual signals that had probably evolved via sexual selection or social selection. And Andy Farkey was thinking about this, and this paper genuinely arose from him sitting there with two model Triceratops, butting their heads together to see what happened. And he realized that if the Triceratops were using their horns in contests between individuals of the same species, then you would expect to see injuries from those horns in particular places on the skull and not in other places on the skull. And we actually have enough Triceratops skulls that he was able to go and assess where the damage on the skulls is. And he found, as you can see, I don't know if you can see my mouse pointer, but he found that healed damage on the skulls of Triceratops is concentrated in the places where the horns would go if they were engaging in contests with each other, and not on other parts of the skull. And that seems to be very strong evidence that Triceratops was indeed using these horns in contests between individuals of the same species, rather than, for example, using them as defense against predators. This doesn't mean they didn't use them as defense against predators, but given the diversity of horns of these animals, it seems most likely that their primary function was as an intra-specific weapon. And another example comes from the group of dinosaurs called the pachycephalosaurs. These had enormously thick, bony skulls, and people had speculated that they used their bony skulls in contests between males, much like modern sheep ram their heads against each other. People suggested that pachycephalosaurs also rammed their heads against each other. And then a paper from these guys that came out in PLOS One in 2013 assessed again where the damage could be found, whether healed or healing damage could be found on the skulls of these animals. And once again, they found that the distribution of damage on the skulls of these animals was consistent with them using their skulls in headbutting contests with each other. So if we look back in the history of life, we find lots and lots of evidence for very similar diversity of weaponry that was almost certainly used in intra-specific contests between animals. I think even the American paleontologists who almost religiously believed that these things were species recognition signals have at least stopped making that argument now. So just a brief foray into dinosaurs to make the point that this has been going on for a while now. And then we need to think about, well, okay, so we've got some groups of animals where we do get these weapons and some groups of animals where we don't. Some groups of animals where males don't compete aggressively to try and get matings with females, and some groups of animals where they do. And that then leads us on to the question of, well, when should it be that males should compete with each other in an aggressive way? And I've got a quote here from a paper by Doug Emlen. Doug Emlen's a professor at the University of Montana. He's really the guy who should be giving this talk. He knows more about the evolution of animal weapons than anyone else. And this is from his monumental review that was published in Annual Review of Ecology and Systematics in 2008. And if any of you are interested in this subject, I'd recommend that you start by reading that. And he talks about how in all the cases where we've got male animals with big weapons, they use those weapons to defend critical resources, and those critical resources would either be females themselves or resources that the females needed to access. All of the time, these resources are limited in space or time, or they're localized so that they can be defended properly. And by defending these resources, the winners of these contests then get lots of access to fertilizations, and these reproductive benefits, if they are high enough, outweigh even the substantial costs of producing and bearing a weapon. So we would predict that we would see the evolution of weaponry in species where there are localized in time and in space, localized resources or localized access to females that can be defended by the males. And we can think about this in a little bit more detail. Another paper, not by Doug Emlen this time, but by Stephen Emlen from 1977, talked about what's called the potential for sexual selection. And they talked about how the distribution of females, what's called the operational sex ratio, that's the ratio of males to females that are actually available for mating, things like population density and mating synchrony should all affect whether or not males evolve to engage in these contests and grow these weapons. And on the right, there's a plot from a paper that I wrote where I looked specifically at the question of population density. And on the graph, we've got the costs of aggression increasing linearly with population density. So as population density increases, you have, as a male, you have to engage in more and more contests. And if each contest carries a certain amount of cost, you would expect that the benefits that you would expect that the overall costs from your aggressive behavior will increase in roughly in a linear fashion. However, the benefits from your aggressive behavior are not going to increase in a linear fashion. And certainly at very high population densities, it's going to be effectively impossible to defend those resources because there will be so many other males. And so the benefits from that aggressive behavior will not increase in the same way as the costs do. And so what we would predict is that we should see aggressive behavior like this evolving at intermediate population densities. At very low population densities, we predict that males should be selected to find females rather than putting all their resources into weaponry to defend those females once they've found them. At low population densities, the problem is finding a mate, not defending a mate. And at very high population densities, the resources, either the females or the resources that the females use, become undefendable because there are so many opponents trying to access them. And so we would expect to see male weaponry evolving at intermediate population densities. And just to show you a bit of data that supports that, we looked at dung beetles, because dung beetles are very, very cool in many ways. We looked at dung beetles at a place in South Africa called Nailsvlei, and we compared species that have horns with species that don't have horns. On the right here is a figure that just shows you some of the diversity of horns that you get in dung beetles. Many species of dung beetle also don't have horns. And we looked firstly at the mean crowding, so the density that male beetles would actually experience when they live at a dung pad and compete with other males. And as you can see on the left, we found that horn species tended to be present at lower levels of mean crowding than hornless species. The very, very common species tended not to have horns. And on the right, we looked at the operational sex ratio, the ratio of males that are willing to mate to females that are willing to mate within the species. And we found that species with lots of males and few females tended to have horns, and species with more females than males tended not to have horns. So once again, we've got strong contests between males here because of the operational sex ratio, meaning they have to try and defend those females. I should say that since this time, we've done another study in Borneo, and we found a rather different relationship between density and the presence of horns in our beetles, but that was in a disturbed area. So in oil palm in particular, we found that horn species tended to be present at higher densities than hornless species. But we think that's because having these contests between males actually allows the species to adapt better to changing environments than not having these contests between males, for reasons that I won't go into. But I just need to say that we've since done another study that shows some somewhat different results, but I've put this in because it makes a nice story. Okay, so hopefully that's given you an overview of the kind of weaponry that we see in these species and the kind of circumstances under which these kind of weapons evolve. As we've said, these weapons are costly, and contests between animals are costly. I think if you look at male red deer, they use something like 30% of their total energy intake for the year during the rut, during the period when the red deer stags are competing over females. So this behavior is extremely expensive. And while you may get fitness benefits that outweigh that if you're a big, tough male with lots of resources, if you're a small, weedy male without many resources, then in some cases, it may not be worth your effort to actually put the resources into weaponry or into aggressive behavior. And that's when we get what we call alternative reproductive tactics evolving. And I'm going to show you some examples of this, some of which don't involve weapons, but they're just so cool that I want to talk about them anyway. So this is my all-time favorite example of an alternative reproductive tactic. This is on the left, marked F, we have a female cuttlefish. In the middle, we have a small male cuttlefish, and you can't see it on the picture, but to the right, there's a large aggressive male cuttlefish. And this, rather than trying to compete with the large male cuttlefish, the small male cuttlefish has interposed himself between the male and the female. And on his right-hand side, he's displaying colors that make him look like a female, so that the male cuttlefish, the big aggressive male cuttlefish, doesn't actually try and compete with him, doesn't react aggressively to him. But on the left-hand side, he's showing male display patterns to this female in the hope of getting a mating with her while this male cuttlefish is distracted. So this is an alternative reproductive tactic. As I say, it's not particularly involving a male, not particularly involving a weapon, but it's so cool that I just love this one. We have examples of alternative reproductive tactics where we have display traits that vary between males. So the famous side-blotched lizard, Uta stansburyana, where we have a genetic polymorphism with males that behave in 3 different ways. I'm not going to talk about that much more, but again, if you're interested in this, I really recommend that you look this up. It's an absolute classic example, and it's very, very interesting. And then we have examples where we have this is another dung beetle species, and we have what are called minor and major males. In this species, Ontophagous nigriventris, you can see that the major male has got a very large horn, and he's gonna fight with other males to get access to females. The minor male hasn't really bothered growing a horn at all, and he's not going to engage in aggressive interactions. He's going to try and sneak around the big aggressive males and get matings that way. And in some of these Ontophagous species, the females dig burrows underneath a dung pat, and the major male with the big horn will sit at the entrance to the burrow, ready to fight any males that try and get in the burrow and mate with his female. And then the sneak males will actually dig side tunnels and bypass the major male completely, and they'll go and mate with the female, and he won't even know they've been there. So we have alternative reproductive tactics. These arise in lots of different ways. So we've talked about genetic polymorphisms, and there's several examples of these from vertebrates, side-blotched lizards, several other lizard species, the bird ruff, where the males come in 3 different types, and that's a balanced genetic polymorphism. We have what are called sequential alternative reproductive tactics, where the males change their reproductive tactics as they get larger. So we see this in many fish species, and also in, for example, some cervids, some deer. So in red deer, the small young males won't interact aggressively with the big tough males, but they will try and get sneak matings with females. And then we have what are called status-dependent alternative reproductive tactics, where the males either follow an aggressive tactic or a non-aggressive tactic, depending on their status. What we mean by their status is how much they have in the way of resources, how big they are, how likely they are to win a fight. And in a lot of insects, these status-dependent ARTs are fixed because the males develop either into these major or minor males, depending on the amount of food they've got available as larvae. And to give you an example of the kind of benefits that we see to small males from not bothering to fight, if a small male doesn't invest the resources that he would otherwise have used for a horn, if he doesn't put those resources into growing a big horn, he can put those resources into other things. And what we find, so this is from a study by Lee Simmons and co-workers, and they looked at a bunch of different species of dung beetle that have these alternative reproductive tactics. And the x-axis here is the standardized testis weight for the major males and the ones with the horns that fight with the other males. The y-axis is the standardized log testis weight for the minor males, so the males that don't grow horns and don't fight. And the line is not a best fit line, that's a line with a slope of 1. And what you can see is that almost all the species in this study have minor males with testes that are bigger than the testes of the major males. So the minor males, rather than investing in weaponry, are investing in testes size. And we know that testes size is very, very closely correlated with sperm competition success in animals. So these minor males, they're taking the resources that they would have used to grow weapons and engage in aggressive contests, and they're putting those resources into something else that will increase their fitness. And in this case, it's investing in testes. A very interesting example from one of these species, which takes us back to what we were talking about with density, comes from this species here, which is Onthophagus taurus. Onthophagus taurus is another dung beetle. It has major males and minor males. You can see this beautiful photo by Alex Wild, who's an American macro photographer here, showing 2 males of Onthophagus taurus, and then this rather less good photo taken by me, showing the heads that I've removed from the bodies of some Onthophagus taurus males. And what you can see is that we've got these minor males which have either no horn or hardly any horn, and then we've got the major males that are growing horns. And whether they develop into minor or major males depends on the amount of resources they get as developing larvae. And if we look at the relationship between body size and horn length, we see a switch point in body size where they start to grow horns and develop into majors. And the interesting thing here is that these beetles were introduced into Western Australia as part of a campaign in the 1970s to reduce the amount of cattle dung that was lying around and forming a breeding resource for flies. So Australian dung-eating insects are not very good at removing cattle dung. So they introduced dung beetles from elsewhere to try and remove it. In Western Australia, these dung beetles are at very, very, very high densities. If you take a spade and you turn over a cow pat in Western Australia, you can find, 500 or 1,000 of these beetles there. And what was found was that after 20 or 30 years of selection at these very high densities, the proportion of males in the Western Australian population that was developing into these major males has reduced substantially. So what you can see is that the switch point here, so the gray dots here are the beetles from Western Australia. And you can see that the switch point where they develop into major males has shifted so that the proportion of major males in the population is much less. And we think this is because the very high densities they're experiencing mean that only the very biggest male beetles in this population can gain a fitness benefit from fighting with each other. And so we've seen the whole makeup of the population shift in response to population density. Okay. Lethal combat. So I've said that most of the time, these contests between male animals involve signaling, assessment, contests that are contests of strength, and that serious injury is rare. So we've got a video here of red deer males, and they're going to start out by fighting, and then another male is going to come along, and you're going to see two of these males engaging in behavior called a parallel walk, where the males walk alongside each other, giving them a chance to assess the likelihood of winning a contest with that male. So we start out, and you can see the two males. You can see they're really engaging in a contest of strength here. They've locked their antlers together. They're not trying to hurt each other. They're trying to push each other, which enables them to assess how strong the other male is. And then there's another male that's just arrived. So these males finish. One of them wins and one of them loses. And then the male that arrived while they were having that contest turns up, and now you're going to see them engage in this parallel walk. So they're walking alongside each other. They're not looking at each other directly, but remember, they've got eyes in the side of their heads, so they can see the other male, and they're assessing the other male. And one of the males is roaring, and this is a signal, again, that they use in interactions with other males. And that the volume and depth of that roar is a signal that you can't cheat. You have to be a big male to make a loud, deep roar. Okay, and they'll continue that parallel walk for some time. And in fact, at the end of that parallel walk, one of the males just walks away. He's assessed the other male. He realizes he's going to lose, and he just walks away. So all of this is a kind of ritualized contest that's evolved in order to avoid these males actually trying to kill each other. These are big, strong animals with pointy horns or pointy antlers. They don't actually want to get hurt during a contest like this. And almost the whole structure of the contest has evolved to enable them to assess the other male so that they don't have to engage in any kind of lethal competition. Okay. In other species, though, we get males that do fight to the death. So these are nematode worms. These are entomopathogenic nematodes. They enter an insect, they kill it by releasing lethal bacteria into it, and then they develop within the body of the insect. And these are two males, and one of these males is now killing the other male. He's grabbed hold of him, and he's squeezing him good and hard, and he's effectively strangling the other male. So why do we get contests? Why do we get these lethal contests in some species when most species try to engage in contests that don't actually end in the death of one or the other, and they try and avoid engaging in damaging fights with each other? So this was modeled by Enquist and Leimar way back in 1990. And what they said was that you can get lethal combat evolving when the value of the resource you're fighting over is greater than the potential value of any resource you might meet in the future. So in the case of these nematode worms, they've entered the body of this insect. They've got one shot at reproducing within that insect. There's going to be a female there, or more than one female there, they're going to try and mate with. If they can kill that other male, they get all the matings in that insect, and they produce thousands and thousands of offspring, and everything's great. They don't get another insect. They're not going to live long enough to enter another insect. And so the value of the resource now is much, much greater than the value of any kind of future resource that they might come across, because they aren't going to live long enough to come across any kind of future resource. And when we look at these lethal contests between males, we find that they're all, or almost all, from invertebrates on ephemeral, on short-lasting, high-value resources. So we see lethal contests between males in parasitoid wasps. We see lethal contests between male larvae within parasitized insects. And if they can kill all the other males, then when the females emerge, they're going to be able to mate with all the emerging females. We see it in these entomopathogenic nematodes, and we see it in fig wasps. If you remember, we saw an example of a fig wasp earlier. We see it in fig wasps, where in some small figs, you get females and males, and a single male can kill all the other males, monopolize the females, and he's then got this fantastic resource that's going to give him very high fitness, and he's never going to be in another fig. He's got one chance and one chance only. So we see lethal combat evolving in cases where we have these ephemeral and high-value resources and short-lived males that get one chance at monopolizing them. Okay. And a final example of this is from this soil mite called Sancassiana bellisii. It's a photo by Jacek Radwan. I'm currently working with Jacek to look at how sexual selection in these soil mites affects their population dynamics over the long run. We're working on some papers, so watch this space. I won't tell you what we found because it's not my data to talk about really. But these males have an alternative reproductive tactic. So the male on the right in this picture is a fighter male. You can see his second to last pair of legs is bigger than his other legs and has got a pointy tip on it. These are weaponized legs that this male will use to kill other males. And then we get males that develop into scrambler males with no weapons. So the male on the left doesn't have these weaponized legs. He's not going to engage in any fights. He's just going to try and find females and mate with them. And the interesting thing here is that the males will only develop into fighter males in low density populations. So there's a pheromone, there's a chemical pheromone that these things release. And the developing males will sense whether they're in a high density or a low density population from the amount of pheromone present. And if they're in a low density population, they're much more likely to develop into a fighter male. And we think that's because they can then monopolize, if they can then kill all the males in the immediate vicinity, they can monopolize access to females. Whereas at high densities, that's simply not going to happen. And the chances of them killing all the other males are going to be very small indeed. So I've gone on a bit longer than I was meant to. Obviously, we started a bit late. So I can either carry on talking and talk about how the diversity of weapons that are used in intersexual contests are used, or we can stop there and just open it up for Q&A, which will keep you on time with your schedule. What would you guys prefer me to do?
Let's try with questions, maybe. If from online audience, from in-person audience, if people can actually, if internet connection allows really to do that.
Okay. So I won't talk about the diversity of weapons. I'll just quickly flick through it and make the point that weapons used in intersexual contests are incredibly diverse, including dinosaur weapons. In antelope and deer, the diversity of horns and antlers is at least partly related to the way that the males fight with each other. And that's a magnificent ibex. In dung beetles, it doesn't seem to be the case, and there's potentially other reasons. And I was going to talk about how in stag beetles, we don't know this, but I was going to speculate a bit with some video of stag beetles fighting. But we'll just go through that. And then I was going to make the point that we have a lot of diversity within species in stag beetles as well. So males with different kinds of mandibles. And currently we know nothing about what these things are actually used for. So if anyone wants to do some research, please go and find out what Odontolabus brookiana males are actually using their mandibles for. And that's it. So if anyone's got any questions, please ask.
Thank you very much. Please, people in person, can you try?
We have question in the audience, so can we?
Okay. Can you hear me?
I can hear you.
Okay, thank you for your presentation. It was very interesting. And I wanted to ask you, is there any data about the who do females prefer more? Males that use alternative reproductive tactics or males that stick with more physical and aggressive approach?
That's a great question. The answer is probably it depends. So obviously alternative reproductive tactics are only going to work if the females are actually willing to mate with the males that are pursuing those tactics. And in some species at least, so in ontophagous dung beetles, the way the males court the females is different from the way that they fight with each other. The females seem to have no preference for horns at all, but the males have a fairly energetic courtship behavior, and both the minor males and the major males do that. And the females seem to have a preference for the males that can do the courtship best, which is independent of whether or not the males are pursuing the alternative reproductive tactics or not. So that's the case in dung beetles, which are obviously the things that I know the most about. In a lot of other species, we don't know. I'm trying to think. A lot of the time, in many of these species, females will mate with males for reasons other than actual preference. So you get a lot of harassment, and females simply mate with a male in order to stop him harassing her. When you have males controlling females and controlling their access to mates, then you get sexual conflict in terms of who's actually going to be fertilizing that female's eggs. And it may well be that they will have matings with other males that may be pursuing alternative reproductive tactics in order to give them more options about which males are going to fertilize their eggs. So it's a big subject. In some cases, the answer seems to be yes. In other cases, I would imagine that females will be mating with males pursuing ARTs for reasons other than their actual mating preferences, if you see what I mean.
Okay, thank you.
One more question, just a minute.
It's maybe some thought I had when you started your great presentation, but can we think about the primates' arms like a specific weapon tool? Like we have some habits in primates that use arms like a weapon, not like for creating something, just for fighting.
Sure. So in most primate species, contests between males are largely going to be mediated by display traits like teeth. So if you think of baboons, which have that very distinctive yawn display where they show each other their canine teeth. And then we have primates that have actual like colored signals, things like mandrills. And in orangutans, you've got the males with those big flanges on their heads that also seem to be some kind of dominance signal. I'm not aware of any primates where the males specifically show each other their arms, apart from possibly arguably humans. And a few years ago, there was a paper arguing something similar to that, and arguing that essentially the human hand was adapted to be used as a fist in contests. I don't think that was very well received. I think a lot of people thought it wasn't very well argued. So probably not in the case of most primates. You could make an argument that in humans, things like bicep size can potentially be used as a signal. But I try and avoid making arguments like this about people because people are just really weird. And human sexual selection is a very, very strange subject that I try and avoid. So in most species, probably not, because they have other signaling routes, possibly in humans and maybe in some other primates, but I don't know. I'm sorry, that's not a very good answer to your question.
Thank you. There is one question in the chat. I will read it out loud, I think, in
Okay.
case if there are more questions in the audience, people have time to think maybe or wait your turn.
Yeah, we have
So,
one more question
okay,
here.
perfect. Should I read then first online question and then
Uh,
I will.
yeah.
Yeah. Yes, I'll read first online question. So thank you for an amazing lecture. You listed a lot of mammals and insects and dinosaurs, but only briefly mentioned birds. They are known for investing a lot into sexual dimorphisms and courtship. Do you have any examples of interesting bird weapons, or there are some other reasons why they do not evolve?
Yeah, that's a great question again. So in lots of cases with birds, we have female choice systems where the males are competing not by trying to beat each other up, but by displaying to females. So the classic example would be something like a peacock or a widow bird, where we have the peacock's train or the widow bird's tail, and the female, or black grouse or sage grouse or something like that, and the males get together in groups in leks, and the females will come to that group and basically assess the males on the basis of their display and kind of go no, 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 yes. And that male will then get lots of matings. And we see this in lots and lots of bird species. And then we also have bird species where we have mutual choice. So if you think of, for example, a puffin, do you know what I mean by puffin? The little birds with the brightly coloured beaks. There's lots of them live near me on Bempton Cliffs in East Yorkshire. Puffins, you can't tell a male puffin from a female puffin by looking at it. And it seems very likely that there's a kind of mutual assessment there where they're essentially displaying quality with these brightly coloured beaks. But it's more a case of males choosing females and females choosing males. But then we also do get some birds where there are proper contests between males. And the main group where we see that is the galliformes. So things like pheasants and chickens and jungle fowl and their relatives. And they have really quite basic weapons, which are the big pointed spurs they grow on their legs, and they use those in fights. And those are proper fights, and they will interact with each other extremely aggressively. What we don't see in birds very much is any kind of analog to the kind of horns and antlers that we see on dung beetles and dynastid beetles, and bovids and cervids. And one thing to remember is that if you're a bird, you've got to fly, or most birds have got to fly, and you probably don't want to grow a massive set of antlers because it's going to get in the way when you fly. Although I have to say, there's been some data come out recently looking at flying dynastid beetles that showed that having these big horns doesn't really affect their ability to fly very much. So to summarize my long and rambling answer, most birds, it looks like it's more a female choice or a mutual choice system rather than males aggressively trying to exclude other males. But there are some bird groups where we do see weaponry and we do see fights. And the main example, as I say, is things like jungle fowl and pheasants.
Thank you. And please, in person, one more question.
Yeah, thank you for your lecture. And my question is about, like, is it dependence between choosing their reproduction tactic, like alternative or combat tactic, between invertebrates and vertebrates, or between size in size of animal, and also between lethal and letate combat tactic?
Sorry, I didn't quite understand the question. Could you ask it again, please?
Yeah. So is it some dependence or relations when between their choosing their reproduction tactic, like for which way will go in this animal, between the size of this animal or between their like it is invertebrate or vertebrate? Maybe some like small creature choose more often their lethal tactic or combat tactic, or like more big
Yeah.
animal will use their.
Yeah, I think I understand. So in insects, we often see these fixed alternative reproductive tactics, like in the dung beetles I talked about. And one thing to remember about those insects is that once they reach adulthood, they don't grow. They don't get any more resources. They get all of their resources as larvae, and then they pupate. So if they choose to grow horns at the end of their larval period, they're then fixed as an animal with horns for the rest of their life. And also, by the time they are entering the pupal phase, they know how much resource they've got, and that's going to determine how big they are as adults. So I think that kind of predisposes them to having these fixed alternative reproductive tactics where they're either a male with a horn or a male without a horn. If you think about some vertebrates, so red deer, for example, they grow a new set of antlers every year, and they shed the antlers after the mating season. And so when they're small, they can pursue alternative reproductive tactics, and they maybe aren't going to put much resource into growing their antlers. And then when they're big, later on in life, they can then grow a big set of antlers and pursue the aggressive tactic. So in terms of that kind of alternative reproductive tactic, I think there is a difference between vertebrates and invertebrates on the basis of how they develop and how long they live. What we also see in vertebrates, what we also see when we are thinking about vertebrate animals, is these genetically based polymorphisms, like in the side-blotched lizard, which are a very different thing from these status-dependent alternative reproductive tactics. I'm not aware of any examples of these from insects, but there are some from shrimps, if I remember correctly. But those are a very, very different thing altogether. And then when it comes to things like the soil mites, the Sancassiana soil mites, as far as I'm aware, that's the only, well, there are several species that do it, of those soil mites, but I'm not aware of any other animals where there's such a big difference between the two reproductive tactics, where one class of male tries to kill all the other males and the other one just doesn't even fight at all. So that seems to be quite a unique phenomenon within the soil mites. I hope that answers your question.
Alexander, do we have time for one more question?
I have one.
And we here in the audience also have question.
Not really. We are past the time already. But I mean, you have to go to lunch to go for next things and workshops. So maybe, okay, well, in person, please, yeah, please continue. Maybe we keep questions and answers short quickly. Yeah.
Okay, thank you so much. Thank you so much. Usually we are talking about male combats and what can we say about female fights? What are the recent and what the differences can we notice between
So
female and male?
another great question, and I apologize for only talking about males. Obviously, as a middle-aged white guy myself, that's just what we do. So we've got several different interesting things that go on with females here. And firstly, we get what are called sex role reversed species. So you may be aware of things like seahorses and pipefish, where the males look after the offspring And the males invest more in reproduction than the females do because they're putting all the energy into looking after the offspring. And we have sex role reversal there where females actually compete for access to males. I don't think they do it with any weapons, but there's certainly, you know, a complete reversal of the normal state of affairs there. We also have examples of females that engage in contests with other females and develop weapons for that purpose. There is one example that I know of female parasitoid wasp larvae engaging in lethal combat inside parasitized insects with other females. And that seems to be to enable them to monopolize that resource that they're in. So there's one example of that. And then we have other examples like there's a dung beetle species called Ontophagus sagittarius, where the females have horns as well as the males. And the female horns are morphologically completely different from the male horns. So the male, I can't remember which way it is, but one of them has horns at the back of the head and one of them has horns at the front of the head. So it looks like they're actually separate evolutionary events. And in Ontophagus sagittarius, the females will fight other females for access to dung. So dung's the important resource for them. They want to make it into brood balls and grow baby dung beetles in it. And in this one species, the females have actually evolved their own set of weapons that are different from the weapons carried by the males. The males compete with each other for access to females, and the females fight with each other for access to dung. So yeah, there are examples where this kind of thing happens in females, although the main, you know, most of the examples we have are from males.
All right. I really encourage people to reach out to speakers. So if you have still more questions, and I think most of speakers are interested and would be really excited to answer your question.
Yeah. Feel free to drop me a line.
Yeah. All right. Thanks everyone. Thanks, Rob.
Thank you all very much.
Pleasure. Yeah.
Have a good one.
You too. Oh, the audience is clapping.